The exchange of oxygen and carbon dioxide takes place in between the lungs and blood. The greater part of oxygen diffuses into the blood, and at the same time carbon dioxide diffuses out. Here the question is where the oxygen would go. Next comes transport of oxygen. Most part of the oxygen, about 97%, diffuses into the blood, is carried by the erythrocyte or red blood corpuscles. Hemoglobin has high affinity for oxygen, and this affinity is increased by the fall in partial pressure of carbon dioxide of blood. At the alveolus in the lungs, venous blood has low oxygen and is exposed to low partial pressure of carbon dioxide of alveolus. Thus, oxygen diffuses into red blood cells and form oxyhemoglobin. As carbon dioxide diffuses from blood to the alveolus, blood partial pressure carbon dioxide falls, increasing the further uptake of oxygen. Oxyhemoglobin remains unchanged till it reaches the tissues where it dissociates readily to release oxygen. Next comes oxygen hemoglobin dissociation curve. The relationship between the partial pressure of oxygen and percentage saturation of the hemoglobin with oxygen is graphically illustrated by a curve known as oxygen hemoglobin dissociation curve. Under normal conditions, the oxygen hemoglobin dissociation curve is sigmoid shape or S shape. The lower part of the curve indicates dissociation of oxygen from hemoglobin. The upper part of the curve indicates the acceptance of oxygen by hemoglobin. Next comes factors affecting oxygen hemoglobin dissociation curve. The oxygen hemoglobin dissociation curve is affected by the factors such as number 1, effect of partial pressure of oxygen, number 2, effect of partial pressure of carbon dioxide, number 3, effect of temperature, number 4, effect of hemoglobin, number 5, effect of pH or hydrogen ion concentration, and number 6, effect of 2,3-diphosphoglycerate or DPG. Next comes Bohr effect, effect of carbon dioxide, acidity and temperature. The Bohr effect is a physiological phenomenon first described in 1904 by the Danish physiologist Christian Bohr, stating that hemoglobin's oxygen binding affinity is inversely related both to acidity and to the concentration of carbon dioxide. The Bohr effect refers to the observation that increases in the carbon dioxide partial pressure of blood or decreases in blood pH result in a lower affinity of hemoglobin for oxygen. Next comes mechanism. Decreases in blood pH and increases in hydrogen ion concentration are likely the direct cause of lower hemoglobin affinity for oxygen. Specifically, the association of hydrogen ions with the amino acids of hemoglobin appear to reduce hemoglobin's affinity for oxygen. Because changes in the carbon dioxide partial pressure can modify blood pH, increased partial pressure of carbon dioxide can also result in rightward shift of the oxygen hemoglobin dissociation curve. The relationship between carbon dioxide partial pressure and blood pH is mediated by an enzyme, carbonic anhydrase, which converts gaseous carbon dioxide to carbonic acid that in turn releases a free hydrogen ion, thus reducing the local pH of blood. Next comes significance of Bohr effect. The Bohr effect allows for enhanced unloading of oxygen in metabolically active peripheral tissues such as exercising skeletal muscle. Increased skeletal muscles activity results in localized increases in the partial pressure of carbon dioxide, which in turn reduces the local blood pH. Because of the Bohr effect, this results in enhanced unloading of bound oxygen by hemoglobin passing through the metabolically active tissue and thus improves oxygen delivery. Importantly, the Bohr effect enhances oxygen delivery proportionally to the metabolic activity of the tissue. As more metabolism takes place, the carbon dioxide partial pressure increases, thus causing larger reductions in local pH and in turn allowing for greater oxygen unloading. This is especially true in exercising skeletal muscles, which may also release lactic acid that further reduces local blood pH and thus enhances Bohr effect. Next comes transport of carbon dioxide. The tissues as a result of metabolism produce carbon dioxide. The partial pressure of carbon dioxide of the tissues is therefore very high, over 46 mm of mercury. The partial pressure of carbon dioxide of the arterial blood in the capillary at its arterial end is 40 mm of mercury. Carbon dioxide therefore diffuses from the tissues to the capillary blood. At the venous end of the capillary, the partial pressure of carbon dioxide of the blood becomes much higher. The blood carries the carbon dioxide in the following forms: A. in dissolved state, B. bicarbonates, C. carbaminohemoglobin compounds, and carbaminoprotein compounds. A. in the form of dissolved state. Because of its high solubility, about 7% carbon dioxide gas dissolved in the blood plasma and is carried in solution to the lungs. Deoxygenated blood and the oxygenated blood carry about 2.7 ml and 2.4 ml of carbon dioxide per 100 ml of blood in dissolved state in plasma. B. in the form of bicarbonate. The dissolved carbon dioxide in the blood reacts with water to form carbonic acid. This reaction is very slow in blood plasma but occurs very rapidly inside RBCs because a zinc-containing enzyme, the carbonic anhydrase present in RBCs, accelerates its rate about 5,000 times. Due to this, about 70% of carbon dioxide received by blood from the tissues enters the RBCs where it reacts with water to form carbonic acids. Carbonic anhydrase is exclusively found in RBCs. All other tissues contain it in traces except stomach and pancreas, which has considerable amount. The most of bicarbonate ions formed within RBCs diffuse out into the blood plasma along the concentration gradient. Exit of bicarbonate ions considerably change ionic balance between the plasma and the erythrocytes. To restore the ionic balance, the chloride ions diffuse from the plasma into the erythrocytes. This movement of chloride ions is called chloride shift Humburger's phenomenon. Next comes is carbaminohemoglobin compounds and carbaminoprotein compounds. In addition to reacting with water, carbon dioxide also reacts directly with amine radicals of hemoglobin to form an unstable compound carbaminohemoglobin. This is a reversible reaction. A small amount of carbon dioxide also reacts in this same way with the plasma proteins. About 23% carbon dioxide is transported in combination with hemoglobin and plasma proteins. Next comes chloride shift. Chloride shift, it is also known as the Humburger shift, named after Hartog Jakob Hamburger, is a process which occurs in a cardiovascular system and refers to the exchange of bicarbonate and chloride across the membrane of red blood cells, RBCs. As a result of catalysis by carbonic anhydrase within the red blood corpuscles, large amount of carbonic acid are produced as blood passes through the systemic capillaries. The build-up of carbonic acid concentration within the red blood cells favors the dissociation of these molecules into hydrogen ions, proton, which contribute to the acidity of a solution, and bicarbonate ion. The unloading of oxygen is increased by the bonding of hydrogen ion, means release from the carbonic acid, to oxyhemoglobin. This is the Bohr effect and results in increased conversion of oxyhemoglobin to deoxyhemoglobin. Now, deoxyhemoglobin bonds hydrogen ion more strongly than does oxyhemoglobin. So, the act of unloading its oxygen improves the ability of hemoglobin to buffer the hydrogen ion released by carbonic acid. Removal of hydrogen ion from solution by combining with hemoglobin in turn favors the continued production of carbonic acid and thereby improves the ability of the blood to transport carbon dioxide. Thus, carbon dioxide increases oxygen unloading and oxygen unloading increases carbon dioxide transport. When blood reaches the pulmonary capillaries, deoxyhemoglobin is converted to oxyhemoglobin. Since oxyhemoglobin has a weaker affinity for hydrogen ion than does deoxyhemoglobin, hydrogen ions are released within the red blood cells. These attract bicarbonate ions from the plasma, which combines with hydrogen ion to form carbonic acid. Under conditions of lower partial pressure of carbon dioxide, as occurs in the pulmonary capillaries, carbonic anhydrase catalyzes the conversion of carbonic acid to carbon dioxide and water. Carbon dioxide is transported in three forms, such as dissolved carbon dioxide gas, attached to hemoglobin as carbaminohemoglobin, and as carbonic acid and bicarbonate. Percentage indicate the proportion of carbon dioxide in each of the forms. When bicarbonate ion diffuses out of the red blood cells, chloride ion diffuses in to retain electrical neutrality. This exchange is the chloride shift. Next comes cellular respiration and oxidation. The ultimate aim of all the respiratory activities is the cellular oxidation, that is the aerobic breakdown of the digested food materials and the release of energy thereby. The breakdown of carbohydrate may be represented by the equation. The complex carbohydrate C6H12O6 is oxidized by oxygen to form carbon dioxide and water along with energy amounting 686 kilocalorie per mole. Thus, within each cell, glucose is first metabolized to pyruvic acid, and then if there is supply of oxygen to carbon dioxide and the water, it is when hydrogen atoms are combined with oxygen that most of the energy is released in the cell. By storing energy as ATP molecules, the cell can utilize this energy for its various activities as needed. From the above discussion, it can be concluded that the oxygen dissociation curve shifts to the right when the blood contains excess carbon dioxide. This is called Bohr effect after the name of the discoverer who discovered it in 1904. Shift to the right also occurs if the pH of the blood is low or the temperature is high. It is now known that the Bohr effect is produced principally due to the lowering of pH by increased carbon dioxide tension. The oxygen-rich arterial blood reaches the capillaries running through the tissues. The tissue is metabolically active, that is, it needs oxygen and its carbon dioxide should be removed. Because of the pressure gradient, carbon dioxide moves from the tissues to the capillary blood, exerts Bohr effect, and oxygen release is hastened. Greater the carbon dioxide tension in the tissue, greater will be oxygen release. That is all for today on chemistry of respiration part 2, Bohr effects and chloride shift. Thank you all.